Hi, this is the second part of changes to DNA, and I'll be discussing primarily transposons in this section. Much of what I'll be discussing you can find in Chapter 13 of the Integrated Molecular Evolution, second edition. As I said in the last presentation, DNA viruses and RNA viruses have some similarities to DNA transposons as well as RNA retrotransposons. There are three general types of transposons and retrotransposons. The first are DNA transposons, which have a transposase gene and often some other genes that aid in the transposition events, as well as short inverted repeats that are used for inserting and excising from a particular spot. Long terminal repeat retrotransposons have a reverse transcriptase, as I've already shown in the last lecture, as well as these long terminal repeats, or LTRs. Non-LTR retrotransposons, at least some of them, have a reverse transcriptase as well as what looks like a poly-A tail. So it appears that they originated from an mRNA that then was copied into a DNA and inserted into the genome. Here are a few more specifics on these different types of transposons. The first one, these DNA transposons move as a DNA, excising or following a replicative pathway. These include AC and DS elements in maize, P elements in Drosophila, TN3 and IS1 in E. coli, TAM3 in Antherinum, and SPM in maize. The second group are the LTR retrotransposons, which have a reverse transcriptase as well as other genes, and these long terminal repeats. They move as RNA intermediates produced by a promoter in the LTR. So all of them have a promoter region in the LTR, and they resemble retroviruses. A few examples are copia in fruit fly or Drosophila, TY in Saccharomyces, THE1 and BS1. The third group also encode a reverse transcriptase as well as other genes most of the time. And they appear to be a processed, or their origin is from a processed mRNA, and they have a poly-A tail already in the gene itself, or in the the retrotransposon. They also move as RNA intermediates, and a few of these are P elements in fruit fly, L1 in humans, and SYN4 in maize. Transposons were first discovered, and the process of transposition was first discovered by Barbara McClintock in the 1940s through 50s. She was working on maize cytogenetics, so she was looking at chromosomes. She also discovered recombination during meiosis in maize. She won the Nobel Prize for her work finally in 1983, and then only lived about 10 years after that, but she won the Nobel Prize for Physiology and Medicine. So she was working on this for a long time, and people actually thought she was a little bit off base, but in the end, once they finally could do molecular work on these changes to the DNA, she was proven right that these were transposition events where these elements were truly mobile genetic elements moving around the maize genome. So she was looking around at different patterns, color patterns, starch patterns, and other things in maize, and found all these strange genetic effects when she did the crosses with certain elements were in the genome. So she could get striped corn kernels, white kernels, things that had speckles, things that had more sugar than starch, more starch than sugar, etc. So here you see some other patterns where you get a blue colored corn all the way to white, which has no pigment, and then you get these stripes of yellow and red, and also speckled kernels as well, all on the same cob of corn. And she was able to map these out and discover some actually predictable events in these maize kernels. Eventually, it was discovered that this phenomenon occurred in other organisms as well, other plants, including pea plants and other types of flowers. You could see the pigments either were striped or speckled or dotted. And so while this wasn't changes in pattern weren't always caused by transposons, it was pretty common in the plant world. She also examined not only many different patterns, but many different genes. So as I said, not only could you see color changes and all sorts of color pattern, but you could see some dimpled kernels and some fully expanded kernels, and this had to do with conversion of sugars into starches. So she discovered that it didn't only affect the color genes, but it affected many other genes in other pathways. So this is just one example of a simple example of how the transposons cause changes in the bronze gene, which encodes for a pigment. So the normal form is capital BZ on the left, and then in the middle, the mutation has no coloration. But then if you have a transposable element in the genetic makeup of this plant, you can get speckled kernels in which some of the cells have the pigment formed and other cells have a disruption of that gene, so there is no pigment formed. So here's one example of one of the crosses that she did. This large C is for the wild type color, and small c is for the mutant with no color. And then you have the dissociator or DS element and the AC, the activator element in the genomes as well. So one has no color, but it has the transposons, and the other one has color without the transposons. And when she did the crosses, the progeny, about half of them were solid pigment, fully wild type, while another half were mainly wild type color with the purple pigment, but then had colorless patches indicating the interruption of the gene that coded for the pigment. And then very infrequently, then she found an unstable colorless, which was mainly colorless with some patches of the purple patches, which indicated pigment formation in those regions. In the case of the colorless regions, those were caused by a disruption of the pigment gene by the transposable element. But in some rare cases, then the transposable element would pop out of the pigment gene and would restore the function of the RNA and eventually the protein so that it could form that pigment. So she did these experiments with ZMAs for many years, for decades actually, in breeding experiments to determine changes in the coloration and the genetic inheritance of these elements. She found many types, but two of the types that she found were AC, or activator elements, and DS for dissociation elements. The AC could move by itself and cause pigment and other genes to stop functioning, while the DS could move, but only if the AC was present. And furthermore, often the DS caused breaks in the chromosomes, which she could observe cytologically. So although this interested her a great deal, and she studied it for a long time, at first it was difficult to decide what was going on. So I mean, if you saw this data, saw these changes, and they were genetically inherited, but they were unstable, what kind of conclusions would you make? So she actually puzzled on this for quite a while and then finally came up with this idea of transposition. These things were moving around, and people have called them jumping genes. And she was ridiculed to some extent by the scientific community, to the point that in the early 1950s, she stopped publishing her work on transposition and transposable elements. But as I said, eventually, in 1983, she was given the Nobel Prize for this work. So what did this mean? Eventually, it was discovered, not by her, but by others, because, it wasn't until the 1970s that molecular biology and ultimately sequencing solved exactly what was going on. So the AC elements had at least two genes, including a transposase, a gene that made an enzyme that caused the movement of these elements. And then they had these short terminal inverted repeats on the ends. And the DS elements had the same ends, but they were missing parts of the middle of the gene. So they were missing the transposase gene as well as other genes. And in fact, some DS elements only consisted of the short inverted repeats on the end. So McClintock found that the AC element could act in cis or in trans. Cis means that it's on the same piece of chromosome as the gene it's going to affect. In fact, in this case, the pigment gene. So it could pop out of that region where it originally existed, move into the gene that it was going to affect and turn that gene's expression off, or it could leave a copy of itself in the original position and still move into the gene and disable it. But she also found that the AC element did not have to be on the same piece of the chromosome, or it could even be on a different chromosome. And it could still disrupt a gene, such as a pigment gene or a starch-producing gene or something like that. And it could either leave a copy of itself in the original position, or that copy could disappear. But sometimes it would leave some of those inverted repeats in those positions where it disappeared. DS elements, on the other hand, remember they were missing part of the center part of the transposon, including the transposase gene. They required that AC was there for their movement. So DS could only move if AC was there, and again, as with AC, it could move into the gene to disrupt it. It could leave a copy of itself in the original position or disappear, sometimes leaving just the inverted repeats. And it could also break the chromosome within the gene that it moved into, and cytologically that could be observed as a broken chromosome. As with AC, DS did not have to be on the same chromosome, nor did AC, and it could affect the same changes to the gene location where it inserted, where it transposed to, including breaking the chromosome. Again, that could be observed cytologically by a chromosome break, which is how it got its name dissociator, for dissociating that chromosome. Rarely, the AC could also move out of the gene to restore the function of that gene, which is when you get those rare patches formed. So by transposing out of the gene could be restored, which was a rare event, and the AC, a copy of the AC would be located either on the same chromosome or a different chromosome. It could also restore a function where the DS element was interrupting the gene, so it would cause the dissociator transposon to move out of the gene, restoring the gene function. As I stated in an earlier lecture, one of these sessions, I indicated that there were LTR retrotransposons that are found in arthropods and other organisms. And non-LTR retrotransposons found in our genomes and other genomes, including those of maize. But there are also transposons found in our genomes as well as plant genomes. So they're quite widespread. One of the reasons that Barbara McClintock was able to find these transposons is they make up a huge proportion of the maize genome. So about 84% of the genome is made up of transposons and retrotransposons, including at least 11 superfamilies. One of those includes the AC and DS elements. And only 5% of the genome is actually made up of mRNA genes and other genes. About 1% is made up of microRNA genes, and about 10% of the genome is other sequences, including introns. So most of the genome is made up of transposons. The positions of the LTR retrotransposons have been mapped on to all 10 chromosomes of maize, and they're located in the central portions of the chromosomes, and you only find the genes, the mRNA genes and other genes, way out towards the ends of each one of the chromosomes, just before you reach the telomere regions. In your own genome, the human genome, there are lines and signs which are non-LTR retrotransposons. There are LTR retrotransposons and DNA transposons. So altogether, those make up 45 to 50% of the genome. Only about 1.5% of the genome is made up of mRNA genes. Another 25% are made of introns. And then the rest of it, which amounts to less than 20%, are other unique sequences and repetitive elements as well. Our genome then is about 45 to 50% transposons and retrotransposons, primarily retrotransposons. And the maize genome is about 84% transposons and retrotransposons. Both of these have been, these transposons and retrotransposons have been very important in scrambling around different gene combinations and making new gene combinations in these organisms. So they've been exceedingly important in the evolution of these organisms with very large genomes. So that's a little bit about transposons, which emanated from viruses primarily, and retroviruses. And next I'll be talking about introns, which also have some similarities to transposons and retrotransposons. And I'll have a little section on plasmids in that lecture as well. So that's the end of part 2 of changes to DNA on transposons. Again, that's from chapter 13 in the Integrated Molecular Evolution, second edition. Alrighty, thanks.